In this video, we will discuss about the role of N-glycan that are present on spike protein on the syncytia formation. When the cleavage of spike protein happens, the S2 sites contain a fusogenic peptide that get exposed and then helps in binding the viral uh, cell membrane of the cell membrane of those cells that are transfected with the spike protein to the other cells. So this experiment is carried out on two kind of cells. First one is the Vero cells, another one is the 293 T cells. So this is the experiment overview. The ACE2 cells that are transfected with ACE2 cells will have the red fluorescence, and cells that are transfected with spike plasmid are green fluorescence in this experiment. And we will observe whether this red cells and green cells will fuse together or not in the different kind of mutations that we have discussed in our previous videos. So this is the first condition, that is untransfected condition. So in this case, the green fluorescence does not contain the spike plasmid, whereas red fluorescence contain the ACE2 plasmid. So we can see that there is no syncytia formation occurs, and these images are taken on from the InQ site. Next, we did it with the parent. This image represents the parent spike protein, that is without any mutation. In this, there is a syncytia formation that can be seen, that is represented by the arrow. So here the red and green fluorescence fuse together. We can see that. So both these cells that contain ACE2 reporter, ACE2 plasmid, and cells that contain this spike plasmid are fused together. And in case where there is no fusogenic peptide, that is shown here, spike delta, we can see that there is less syncytia formation that can be seen here. So this is aligned with our understanding that fusogenic peptide helps in syncytia formation. And these are in the panel B. We can see the results first of the different. You can see the results of the different mutation. So the left hand side axis represents the measurement that is done on the InQ site, and we can see that the signals from this N801Q mutation and this N61603,657 mutation show slightly decreased signals. So this represents that this mutation helps in reduction of the syncytia formation by 10 to 20%. Whereas when they did the flow cytometry analysis, they can see that there is not much change that they can observe in the flow cytometer. In flow cytometer also they measured both the green signals and the red signals that are available from the cells or from the event, we can say that. And when they did the same experiment with the Vero ACE2 cells, they see the similar results, that in case of InQ site measurement, the mutations of the spike protein, that is N801Q and N61603,657Q, has slight decrease in the signals compared to the other mutation and also compared to the parent. Whereas when they did the flow cytometry analysis, there is not much difference that can be observed, because the InQ site measurements are, we can say that, more precise, whereas flow cytometry measurements are more like we can say that, to measure the amount of signals for a large scale experiment, whereas InQ site measurements can be done for a small scale experiment. So the minor changes are more visible in the InQ site compared to the flow cytometer. It is the reason we can see the minor changes in the N801Q mutation and N61603657 mutation, and these results are observed from both the kind of system. So from this they have concluded that this N801Q mutation and this N61603657Q mutation helps in reduction of the syncytia formation, and these glycans might play some role in the syncytia formation in the SARS-CoV-2 spike protein. So if you need to understand, I hope you have understood something about the syncytia formation and role of N-glycans on this syncytia formation. There is not that much role involved, as these signals are only reduced by 10 to 20%. If you need to understand this thing in details or this topic in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.